Hi, everyone. I'm not Bishvu, and we're not in alphabetical order because we are a conference on evolution and medicine, and she has a medical conflict in her family right now, and she can't come. And very kindly, Kathy allowed me to jump in for her. So Bish is an undergraduate student in my lab who's done tremendous work, computational analysis, studying the patterns of evolution across vertebrate sex-determining genes. So we have some pictures here of mammals, birds, some reptiles. There's snakes and turtles and lizards and crocodiles and fish. And next slide, please. I was told not to touch it, but now I will. Okay. Next. All right. It turns out the sex-determining mechanisms in these species are very different. So you can have X and Y chromosomes. In mammals, it actually should be ZW in birds. In reptiles and turtles, we have temperature-dependent sex determination. XYZZ. In fish, you have behavioral and environmental sex determination. But it turns out there's this set of genes that we show here are conserved in sex determination regardless, in the pathway regardless of whether it's chromosomal or temperature-dependent. And so what she's done is looking across the genital ridge development by potential gonads, looking at the evolution of these genes. We show extremely high conservation of these genes across all species. And it's fascinating that these genes are picked up in sex determination regardless of which mechanism you're using. Thank you.
Nope, the other way. There you go.
Okay, just push that one more.
This one? Okay. Hi, everybody. I'm Catherine. I'm a grad student at University of Minnesota. And as you can see from my affiliations, I'm with the Department of Ecology, Evolution, and Behavior, as well as the Minnesota Center for Philosophy of Science. So I'm a biologist and a philosopher. So I wanted to use this time to introduce who I am a little bit more, as opposed to a single experiment. So as a philosopher, I am working on how the chosen methods that we use in cancer research affect our conceptual models that we use. And one instantiation of this is the use of microbial experimental evolution, with which I ask how can we use microbial experimental evolution to understand cancer. And my answer to this is that we can understand the evolutionary, ecological, and developmental dimensions of causality in cancer through these experiments. As a biologist, I work in an experimental evolution lab with the multicellular yeast system, and those are two pictures of our yeast. And I ask questions about the causes and consequences of modularity. And so the results that I get from my experiments, I can use to inform a new conceptual model that I'm developing, one that embraces the dynamic relationships between levels of organization in cancer. Thanks.
So hi everyone, my name is Marta and I'm a graduate student at McMaster University, and the title of my poster is Writing About Complex Problems Induces Sadness in Analytical Rumination. So, well, I totally missed that. So research suggests that writing about one's personal problems and complex life stressors can be an effective treatment for depression. So our studies examine why this might be the case from an evolutionary perspective of depression. And some of you might be familiar with the analytical rumination hypothesis, but just in case, I'm just going to explain it. So this perspective suggests that depression is an evolved response to complex problems, and that symptoms of depression, such as sadness, anhedonia, changes in concentration and sleeping, promote a systematic style of processing that facilitates problem solving. And it's referred to as analytical rumination. So we hypothesize that writing about complex problems promotes long-term problem solving by first increasing sadness and analytical rumination. So next slide, please.
Can you push the button?
Oh, sure. So to explore this idea, we conducted 2 studies testing a variety of writing conditions. And we found that participants who wrote about their problems reported increases in sadness and analytical rumination during writing as compared to participants who did control writing tasks. As well, we found significant associations between sadness and analytical rumination across all writing conditions. So these findings suggest that writing as a treatment for depression might work by promoting the natural course of depressive emotion and cognition rather than by opposing it. And that's it. Thank you.
Hello, my name is Alejandra, and in the UK, as in many other countries, migrant groups have longer duration of breastfeeding in general. However, this trend tends to erode over time in younger generations. Most of the previous work has focused on acculturation and socioeconomic reasons for this trend. However, I ran an exercise using life history theory principles and reproductive ecology principles to play with a couple of variables that are relevant for fitness, which are infant survival and reproductive opportunity, always for this exercise from the perspective of the mother. And running in it two scenarios, one, high pathogen risk, low energy availability, common to many countries where the migrants come from, and another, low pathogen risk and high energy availability, which represent most of the host countries. What I found was that in scenario one, the longer breastfeeding is benefited, whereas in scenario two, shorter breastfeeding duration is favored. And then I compared breastfeeding data that I have from a migrant study of Bangladeshis in the UK, and what I found was that the predictions did match their data, or the other way around, where women who were born in the UK tend to have shorter breastfeeding rates than those children that were born in Sylhet, even within individuals who gave birth in both places. The public health message, I guess, is to continue doing promoting initiation of breastfeeding, but also pay a lot of attention to safe early weaning practices and family planning for these women who might have increased reproductive potential. Thank you.
Canine distemper virus is closely related to human measles, and recently it's begun to jump species. This includes to non-human primates. Of course, this raises concerns that we could be next. Codon usage is an important factor that influences the ability of viruses to adapt to new hosts. So we have hypothesized that optimizing the codon usage of the virus to that of the host would affect its pathogenicity and its ability to jump species. The question for this particular study was if codon usage bias would affect protein expression from these two viruses in human cells. So canine and human codon usage biases are extremely similar. If you increase the optimization of the canine distemper virus to the canine codon usage bias, it will also increase it to the optimization of the human codon usage bias. There are effects of both codon usage bias optimization and suboptimization on a viral protein expression, and these are completely independent of the viral infection. They vary by gene, and they differ between both of these viruses. Some of the differences that we've seen are in proteins that are involved in attaching to host cells. These, specifically, this would be the H protein. This, combined with the research of others, suggests that the H protein in particular may be very important in the ability of canine distemper to jump to other species. Of course, there's much more to the story, and to hear the rest of it, including the actual definition of codon usage bias, you should come visit me at the poster. Thank you so much.
Hi, my name is Beate Peter. I work at ASU as of recently, and I study speech. Speech is one of the most underappreciated human abilities. Just think, once you go order your favorite beverage, by the time you say a double non-fat tall mocha, you will have activated all the muscle groups necessary for speech, and you will have produced all the phoneme classes of English. It's amazing. On this picture, on the first slide, you see a photograph of an actress in a MRI machine, and she's producing the phoneme L there, and she's doing all the things necessary for that particular phoneme. She's adducted her vocal folds to make sound. She's closed off her velopharyngeal port for oral resonance, and she's opened her mouth. It's amazing. So next slide. Most children acquire their speech sounds of their ambient language by age 4. That doesn't happen for all children. However, children with speech sound disorders struggle with making speech sounds on schedule, and the most severe form is childhood apraxia of speech. These are children who make very weird speech errors. They have limited consonant inventories. They even make vowel errors weird. Some of them leave off consonants in the beginnings of words. And CAS runs in families, and we think it has a genetic component. Childhood apraxia of speech, if we knew what causes it, we could identify babies at birth and do something instead of waiting around until they're 3 or 4 and then start intensive therapy. Last year, we found a deletion of a blood gene in one child, the BCL11A gene on chromosome 2, spontaneously deleted. The child has severe apraxia of speech. So here today on the poster, we report on 2 multi-generational families, also with segregating apraxia of speech. And we did not find the BCL11A deletion, but we found a range of novel markers. So if you want to know more, come to the poster. And if you know anyone who would like to postdoc or be a PhD in our lab, let me know. I have business cards. Thank you.